guess in an era of more targeted therapies, thinking about the complexity of the microenvironment might make it more difficult for us to know that a particular agent would work in a particular way. But we are beginning to see quite exciting data from targeting the microenvironment that we were talking about this morning.
Yeah.
And of course that's nice because the work has come from the understanding the biology of what's going on there. So an approach of looking at the histopathology, the molecular genetics, an understanding of the interactions with the immune systems, understanding what those are, and then having rational target introduction. Now again, of course, many of these have also showing great promise in solid tumors as well as in malignancies. So among the things that we talked about this morning, what kind of piques your interest in terms of looking at the clinical results that are emerging with some of these newer agents?
Yeah, so sure, you've raised some very good points. It's actually, I'm sure, a source of frustration for yourself, as it is for me, that our understanding of the composition and the biology of the microenvironment is in many ways significantly better than what people have in melanoma, kidney, lung, etc. All these other cancers where the whole concept of cancer immunotherapy has emerged, and in fact the journal Science called it the breakthrough of the year last year. And yet our understanding in the lymphoid cancers has, I think, actually leapfrogged that now. So we have the actual alterations, be they translocations, be they other mechanism mutations, that lead to deregulated expression of certain molecules for which we already have now targeted agents. So it's a bit frustrating that we read the New England Journal to some extent, and we read about antibodies to PD-1 or to its ligands in lung, kidney, etc., where the diseases for which we have compelling biologic data are Hodgkin lymphoma, primary mediastinal large cell lymphoma, for where we know those mechanisms are critical oncogenic drivers. That's the subsets of disease for which we could already now be actually entering trials with those targeted agents, where from a biological point of view, it's absolutely a no-brainer.
Oh, totally. I mean, I share your frustration. I've been banging on the door of those companies to try to get hold of those agents for clinical trials in lymphoma for many years now. But finally they're appearing, and we're starting to see indeed promising results in that setting. The other interesting agent, of course, that we kind of have focused more on hematologic malignancies, and of course it had its mainstay in myeloma first, was lenalidomide, which, although in myeloma appears to be directly cytotoxic to the tumor cells, in the lymphomas appears to be having a much more pleiotropic effect, largely focusing on the immune environment, and perhaps mimicking in some ways what some of the other antibodies do in terms of down-regulating expression of some of these molecules. Now, we're of course going to see in more detail from some of the later clinical sessions, but of course very exciting data emerging with the use of lenalidomide and rituximab, either alone or in combination with chemotherapy, both in follicular and in diffuse large B-cell lymphoma. So of course that raises the issue about, should these agents be used in combination with chemotherapy, or are more targeted therapies going to replace conventional chemotherapy for these diseases?
Sure, so of course you're asking the wrong guy, because I'm the guy doing the pathology and the biology, but I can weigh in with the comment. So I think we are not far away from, in certain lymphoma subtypes, so not moving completely away from a paradigm of multi-agent chemotherapy, but of a rational combination of agents where at least one of them is directly impacting the crosstalk between the tumor and the microenvironment. But I think what will be absolutely imperative going forward is that we build in studies, particularly correlative studies, that allow us to understand, for instance, the absolute direct action of something like lenalidomide, not so much on the tumor, but in fact on the relationship between the tumor and the microenvironment. So you yourself, again, have generated data as far back as 2009, looking at the synapse in follicular lymphoma and how lenalidomide can overcome that defect. But to really collect biopsy material on treatment and really try to understand some of the nuances of what's going on with that crosstalk, I think will be critical, because although it will certainly inform on the biology or the impact of lenalidomide and the mechanisms by which it accomplishes its, its efficacy, but it'll also inform on other targets that we could potentially also go after, using potentially agents that also can target the microenvironment.
Now, for the last 25 years, you and I have been at many meetings talking about the promise of immunotherapy, and until now it's always been promise. Many of the original promises, things like tumor vaccines in follicular lymphoma, when it came to randomized clinical trials, showed really no efficacy whatsoever. What do you think is different now compared to our false hope for immunotherapy for the last 25, 30 years?
Well, I think a fairly detailed understanding of the biology and the mechanism certainly informs on that. I think that those strategies in the past that were more patient-specific, although certainly CARs have all the hype these days, I think that a fundamental understanding of the biology is beginning to inform on, and even the agents that are again being talked about in Science magazine as the breakthroughs of the year in cancer immunotherapy. I think we suffer from a problem in the lymphoid field, like you mentioned. You and I have been advocates that that's the patient populations we should be targeting, but unfortunately we deal with relatively small numbers that don't look attractive to the pharmaceutical industry in general terms, and I think it's just been a tough sort of road to hoe to get those agents into clinical trials that involve lymphoma patients. However, I think that the data are there that suggest that those agents are going to be very efficacious. And I say that predominantly because, as we said earlier on during the session, that we have the credible data of genetic alterations that directly tell us that those targets are fundamentally important to those tumors. And so as a growth and survival advantage, those tumors rely on that. And if we can target them with specific agents, they ought to work very well, alone or in combination.
Okay. So to conclude this, for those hematologists, hematopathologists that couldn't be with us today, what's your take-home message from that session, succinctly?
So when you look down the microscope, don't just look at the tumor. Remember that there is a microenvironmental niche there, and it's important. And to think about any attempts we can do as colleagues to enhance the research activity in this area so we can get some of these agents into the front line for people with lymphoma.
And I guess mine would be that we've got very exciting clinical trials emerging looking at this area, and that clinicians should think about referring their patients in for the appropriate trials so that we can get the answers as to whether these agents are really going to fulfill their promise.
And then we can leapfrog the epithelial people.
Absolutely. Thank you very much.
You're welcome. Thank you.